Okay, so thank you for staying back. I think the dinner, I'm hoping Arijit's dinner invitation was strong enough for you to actually stay back. So, so right now so this session is very unique. In one sense, the first speaker is Arijit's student. The next speaker is Arijit is the student. So first is Nikhil. So Nikhil will talk about beyond observation and towards solutions. So let's see what solutions he has to present.
Yeah. Good evening, everybody. Welcome back after the tea break. I'm Nikhil Chukla, a third-year PhD scholar working with Dr. Ajit Samal in the Computational Biology department at Institute of Mathematical Sciences. So today my talk is going to cover beyond observations towards solutions. So what are these observations and what are the solutions? As Professor Himanshu has asked, let me cover it in the next 10 to 15 minutes, if I hope so. Right? So environment and health. So we want to try to understand what are the impacts that the environment has on health. So if you see air pollution, we say air pollution causes cough and it impacts our health, or we have food that causes food poisoning and we are having diarrhea, or we are affected by viruses like corona, having some lung infections, or we are using some cosmetic products and having skin rashes. So all of these, in some form or the matter, affect our health. And that doesn't depend on what our biological makeup is about. So same is seen through this graphic here. If you see so, only 10% is actually made up by our innate immune system, if you say so. The rest 90% is actually decided by other factors such as societal factors and environmental factors. So indeed, environment plays a major role in the upkeep of our health, and therefore we would like to understand this environment and how can we protect ourselves from this environment. So to understand this environment, we should understand something like how is environment affecting us. So there's a concept of exposome that is an indicator of the environmental effect, right? So exposome encompasses all life course exposures to any factor that is from our immediate environment, be it mental stress, be it loss of sleep, be it insomnia, or any other chemical form like air pollution, pollen, for instance. Some of us have pollen allergies and all. So this exposome is a measurable form that we can understand how to look at the environment to see how it affects our health. In 2005, there was this paper by Wild. In 2005, it's a perspective by Sir Christopher Wild, where he says that exposome is a complement of genome. As we've seen in our previous slide, only 10% of the health is dependent upon genome, and the rest is upon the environment. So similar is this function. So if we consider health as a function of genome, it is false. Health is a function of both genome and exposome. So then, in particular, our lab is interested in this chemical exposome, so potentially harmful chemical spaces that can affect our humankind. So what is this chemical exposome? These are environmental chemicals that contribute to various disease burden in our human body. And it is of increasing interest over the past decade. So this is a paper from 2010 where they have looked at how the endocrine disrupting chemicals or neurotoxicants can affect our human body. And finally, there's a review paper from 2020 by Barabasi's group where they have tried to link the chemistry and biology through exposome research. So this is what I'll be talking today. So this is we are trying to link the chemical exposome to our health state, right? So in particular, our lab uses these toolboxes, as we say so. These are 5 unique different toolboxes that were developed and adapted to the study of chemical exposome and how it affects our health. So the first is the curation of chemical atlas. So this has been done extensively by our lab by one of our senior PhD students, Dr. Janani Ravichandran. She has finished her PhD last year. So she has extensively worked on it and systematized a manual workflow with which we can extract data that is already buried in our published literature. So let me start. So for this, to show an example of how we build our chemical atlases, let me take a recent publication of ours. I was involved in this. So all of you know that this is a skin depigmenting disorder called vitiligo. So it happens through autoimmune processes. So the environmental exposure to certain chemicals can cause pathological killing of the melanocytes, and this causes vitiligo. And till date, people have thought this was based on the genetic makeup of the person, but it also turns out there is an environmental factor to it. And we had created the first database that catalogs the chemicals that can cause vitiligo. So how did we go about it? So we had a standardized workflow developed by our senior Dr. Janani. So what we have done is we have extracted data from PubMed. We have systematically and manually passed through each and every information of this literature and extracted the information on 113 unique chemical identifiers after standardizing and cleaning all the data, right? And next, we were able to also extract the various, so if you see various levels of, so there's a patch test evidence exposure. So what it essentially says is they have actually tested it on human patients or small doses of these chemicals and seen that depigmentation occurs. So this is a direct in vivo linkage to these chemicals to cause vitiligo. Then we have something called no patch test observation, where people have objected to getting these patch testing done on the skin, but they have biopsy samples to show that the chemicals, the exposure to certain chemicals has caused the loss of melanocytes. And finally, there are some in vitro lab-based studies that have looked at enzymatic kinetic assays. Then we have categorized them across various exposures that are reported and based on various environmental sources. And all of this we have curated in an online database called WiseKB. It is available to view on our website. It is available at cb.imec.s.in/WiseKB. So all the chemical data and the chemical information has been cataloged on this website. The link to the website is also on the poster outside, so please do attend and scan the QR code to immediately go to the website. So next, we have collected this information. So what analysis can we do about this? So first immediate analysis is it actually present in our day-to-day products that we are getting exposed to? So we have used something called the Chemicals and Products Database. This is unfortunately a US-specific database. We couldn't find any India-specific databases to identify what are the regular commerce products in our daily life. So here we see that many of the chemicals are found in personal care products, home maintenance and cleaning and products. So usually the Lysols, or I'm not naming any particular brand when I say Lysol. I just mean that it is a room cleaner in the office, home cleaner, or personal care products as in face washes, the skin care regimens, and also they're found in bathroom fresheners and fragrance and biocides, right? So we are actually getting exposed to these chemicals on a regular basis, right? But not all of us have not all of us have this vitiligo, so there is some genetic aspect also playing into the effect. So we have clearly not yet understood how it actually happens, but we are trying to focus on the chemical aspect of it to do further analysis. Then we also check, are these chemicals regulated any skin sensitizing potentials? Skin sensitizing chemicals, do they have they been tested for the skin sensitizing potential? And turns out they are very less. So hardly 40 of these chemicals have been known to be causing skin sensitizations, and the rest are still in the out and unknown chemicals. So we have created something novel in their database that people can look at and academically try to support this data and for future purposes. We have also compared it with our existing chemical atlases that were developed by Dr. Janani. So we have compared it with endocrine disrupting chemicals, neurotoxicants, chemicals found in children's products, and also chemicals found in human milk contaminants. So chemicals that are human milk contaminants, and we see there is a very low overlap. So this is a novel chemical atlas that we have generated, and we have extensively cataloged all the information related to these chemical databases. Next, we try to use a toolbox called cheminformatics. So similar to bioinformatics, what cheminformatics does is we take the chemical structures as sequences, like a identifier of these chemicals, and try to understand how they are related to each other. So the first one is about this one is about the chemical similarity network, where we have considered the similarity of, if more than 50% similarity, then that exists, and each of these nodes are essentially chemicals. Here what we have done in this analysis is that we have captured the core part of the chemical, that is a closed loop structure. So anything like a benzene structure or a linked ring structures, they are called scaffolds of a chemical. We see that of these 113 chemicals, there are 56 unique scaffolds. So it's a highly diverse based even on the chemical skeleton of these chemicals, we can say so. So next, we have further also compared these scaffolds with other skin-specific databases to see if the scaffolds have been captured, if not for the chemicals. And we see, this is a scaffold diversity plot, and we see that WiseKB is the most diverse among all the chemicals that are prohibited for use on skin products. And this actually doesn't give much information, but what gives more information is this thing. So if we see 31 of these 56 are actually unique to the chemicals present in vitiligo. So essentially, this contains a lot of novel data to analyze for future purposes. Right? So the red one is actually vitiligo in our WiseKB database. Finally, what we have done is we have leveraged omics and strategies to try to see they're chemically diverse, but do they show similar biological effects? So here we have used something called the LINKS. It stands for Library of Integrated Network-Based Cellular Signatures. So what essentially it does is it's a consortium by National Institute of Health in the USA where they've tested roughly 30,000 compounds on various human cell lines, and they have extracted transcriptomic signatures of the 1,000 key genes that they say so. We have extracted the data from their web server, and we have seen an overlap of roughly 20 of our 113 chemicals which are present in this database. We have extracted the transcriptomic signatures. We have looked for significantly differentially expressed genes in log2 fold change value and significance value of 0.05. And we use standard tools to do enrichment analysis for differentially expressed genes, and we see that it is found as expected. Many of the chemicals trigger signaling pathways in the human skin cell samples, and the reactome shown that it actually targets the receptor tyrosine kinase pathway. So this is the story on the YSKB database that was recently published in January in the journal Science of the Total Environment. And so next, I will go on to the next framework that we have used in our study, which is the adverse outcome pathway framework. So this is a pretty novel framework in the sense that it is envisioned to help with the 21st century toxicity testing program. So it was envisioned way back in 2007 by this US National Toxicology Program, where they say an adverse outcome pathway is a model that identifies the sequence of molecular and cellular events required to produce a toxic effect when an organism is exposed to a substance. So in simpler words, what we can say is that it tries to identify what is the biological process in which the chemicals effects can be seen at various biological scales. In a sense, how it interacts at the biomolecular level and how does it translate to the cellular response, the tissue response, the organ response, and finally, the organism response. So the organism response is something that is of regulatory concern, and we can use this framework to understand or prioritize more chemicals based on these kind of understandings of chemicals we have. And it will also be understood as a modular aspect in the sense that each of these events are called key events. So this is a molecular initiating event, this is also key event, and this is an adverse outcome, is also key event. And they're linked to each other to something called the key event relationships. And these key event relationships have various taxonomic applicability, life stage applicability to also to chemical regulations at various stages of life, or even sex-specific understanding of various pathways. So it essentially happens that if a toxicant is present, it kickstarts this domino effect in the sense that it tilts the molecular initiating event, and there's a domino effect until the larger individual scale and now even the population level. Right? Next, what we see is that we can leverage this information, this individual pathway information, and link it with other existing pathways to look for something known as adverse outcome pathway. So what are these adverse outcome pathway and how is it useful for us? So before I talk about how is it useful, let me first explain how we get about adverse outcome pathway network. So if we say there are two AOPs, so the MIE1 to AO1 and MIE2 to AO1, where there is KE2 is common within the between the two AOPs. So here we can link the two to look something like this. So it's a connected piece of network where we can analyze. So this can be like a critical node which is both present for both pathways. So we can read. we can analyze such networks based on various either stressor specific pathway to look at what MIE can cause. MIE can cause multiple AOs, or we can even look at disease specific. So we can fix on the AOs and look at what all MIEs can actually link to the AO. So in our study, what we have focused on is inorganic cadmium. So inorganic cadmium is essentially, cadmium is a heavy metal. It is known for, it's highly toxic. It's an ecotoxicant as well. Not only does it affect humans, but it also affects various marine ecosystems as well as plants and animals as well. And there's a recent article in 2023 that said they were able to find cadmium in dark chocolate. The chocolates that are it has found its way into the cocoa beans, and the cocoa beans have found its way into the market. So there is increasing risk of cadmium in our day-to-day life. So we wanted to understand what are the possible ways in which cadmium can cause toxical effects in humans as well as ecological systems. What we have done is we have leveraged something called as AOP Wiki. So AOP Wiki is an online resource that catalogs all adverse outcome pathways that have been developed all over the world. It is a continuously updated resource, and it is updated regularly on a day-to-day basis. So it is highly dynamic. So what we have done is we have taken a snapshot of this resource. We have extracted all the data, all the KE data, the KER data, the applicability data for each of these AOPs. We have cleaned the data to get something known as high confidence AOPs. So why do we need this high confidence AOPs? As I told you, it's a living data that can be multiple AOPs that are incomplete, right? multiple AOPs that are not annotated well for their molecular initiating event or their adverse outcome. So finally, we did a novel way of integrating various resources into identifying what are the key events that can be associated in organic cadmium. So here we used 5 different resources to link to the existing keys. So the first one is naturally the AOP wiki, where there were 2 AOPs within AOP wiki that already had the prototypical stresses to be cadmium. So we have considered all the keys to be associated with cadmium. Next, we looked up CTD. So CTD is Comparative Toxicogenomics Database. So it's the largest online database that catalogs chemical gene, chemical disease, and chemical phenotype relationships from the published literature as well as various other online databases. And we have created something called a CGPD tetramer. So it is essentially chemical gene phenotype disease tetramer based on various curation levels that we have put various cutoffs on. We have kept it very stringent to understand only experimental information that we wanted. Then we looked up ToxCast. So ToxCast is another US EPA project on identifying. So this helps us identify potential molecular initiating events. And then Reduct is our own in-house database on endocrine disrupting chemicals, and NeuroToxKB is a in-house database on possible neurotoxic endpoints. So we were able to leverage all of that, and through our integrative approach, we are able to identify these 30 AOPs that are linked to cadmium, based on various other criteria as well. I would suggest all of you to visit our poster to understand what criteria we have used. For the lack of time, let me just say these are the 30 AOPs which we have considered having high coverage score. And we see there is one huge network here. So essentially this network is AOPs are nodes, and the edge exists if they share at least one KE between themselves. So here, this is a largest connected component of 18 AOPs. I'd like to point out that this AOP network contains all AOPs that we have identified to be associated with cadmium through our integrated approach, and this doesn't contain any AOPs from AOP wiki. So we have found something novel, something new that can be associated with cadmium. Then we went on to build a directed network for this approach, and we did 2 case studies that we quickly summarized the 2 case studies. So first case study is on preeclampsia. As you all heard the first talk of today, where Professor Rahul Siddharthan has said that preeclampsia has a risk of highest mortality in pregnant women. So we see that cadmium actually can induce preeclampsia. So how does it do that? So it does it through activation of AHR. It goes through this process of dimerization of HRARNT, reduce VEGF protection. It flows down to placental labyrinth vasculature morphology, and which leads to increase in placental insufficiency. So as Professor Rahul has told today morning, that placental abruption is one of the key features, and preeclampsia is just a specific outcome of this placental insufficiency. Second, we have also looked at the aquatic organisms and how they can affect aquatic organisms. In a similar pathway, it starts at activation of AHR, but this time it goes to something called as increasing COX-2 gene. So COX-2 gene is involved in development, cardiovascular development in fishes. So this increase in COX-2, actually what it does is it creates abnormal cardiovascular development, which leads to high mortality in these aquatic fishes. So this is a recent publication, which was published again in the Science of the Total Environment. And so this is something novel that we have done. We've actually linked cadmium to 2 conditions which we usually observe, but this is a potential toxicity pathway in which they can manifest in these 2 conditions. So to conclude, one, I have talked about a new chemical atlas called WISE-KB, and we have used various cheminformatics tools to understand this chemical space. Second, we have shown the use case of AOP network and how to understand this AOP network to build it to this. And overall, we think that all of this can lead to One Health, in a sense that we can link the chemicals. If we are able to control the chemicals that affect our humans as well as our environment, we can create a better society for all of us. So to end, I would like to thank my guide, Professor Ajit Samal, for giving me the opportunity to talk here, and the various collaborators from lab, and also the collaborators from National Center for Coastal Research with whom we have done the cadmium work. They are sitting in the audience right now. And finally, IMSC and HBN and DAE for the generous funding. So I would like to end with this quote. So human beings are not in control of the nature, but simply one of its parts. The survival of one depends on the health of all. Thank you.
For the first project, very cool stuff. Have you tried using machine learning to predict new chemicals that might induce vitiligo, because you have a training set now of this database?
Yeah, we can potentially do that in the future. Our initial first case was that training data set was itself not present. So we were trying to build the training data set first, and try to categorize it, saying that it is indeed novel and it is indeed useful to understand that. And still we see that only hardly half of them have experimental evidence. So it still begs to differ that we get experimental proof for the remaining of the chemicals before we continue on with the machine learning programs, as you suggest.
Hi, two quick questions. One is, how did you classify these scaffolds? What was the cutoff? And you showed this scaffolds, chemoinformatics toolbox, right?
Yeah, this one.
Yeah.
Yes, yeah.
So how did you classify these scaffolds?
So this is a very simple definition. So we use something called as a Bemis-Marco definition for a scaffold. So essentially what it does is it tries to find the closed ring structures present in any chemical. So if it of course uses information on linear compounds, we use closed ring structures to identify the scaffolds. It leaves out all linear compounds, and if anything is close, it considers that to be a scaffold of the chemical.
So why I was asking, because these are looking very nicely different structures, but if you look into the chemistry,
Yes.
and you will find, there is an ethylene, and then you convert one carbon to nitrogen, and it is a completely different scaffold.
That's true. That's true. Yeah.
But this kind of classification may not capture that small change.
Yes, yes.
So that's what. And the second one was the links data that you have used,
Yeah.
right? So links 1000 or something.
Yes.
So that is mostly trained against the cancer cells.
Yes. So human cells that can be plated on a tissue, on an animal.
Okay, and this is
These are immortalized cancer cells. Yes.
immortalized cancer. Yeah, cell lines.
Sorry, human cell lines, yes.
Okay, so just wanted to see, like, how did you extrapolate the effects of those chemicals into the other chemicals?
Okay, so the basic premise of this links data set was to do something called the connectivity mapping, where they were using it to look at different similarities between various drugs they were using. So we thought maybe we can see for similarities between various toxicants. So we have not exactly probed into the individual genes as you suggested, but
Connect with these chemicals.
yes.
Maybe you can train a model or something. So
Yes, we'll keep it in mind.
that could be used.
Sure. Thank you very much.
The first part of your talk, you talked about these unique scaffolds. So
Yes.
what's known about the targets of these scaffolds? Are they clustered around a set of proteins, or they are diverse in nature?
Targets meaning how?
In targets by which these are bringing about.
So as you see, many of them we were not able to capture from various studies. So many of these chemicals are new. In a sense, they have not been studied against any existing databases. Even this links assay, we are only able to find 20 of our 113 chemicals. And based on that, I have made my observations that they are triggering receptor tyrosine kinase pathway, but it still begs to us to experiment on the remaining 80, 90 chemicals to understand what exactly is happening. So the information is not available on these chemicals. So these are, that's the thing.
Interesting. Thank you.
Thank you.
Yeah, Nikhil, a lot of work, different things that you've tried to put together. If you go to your two use cases of cadmium toxicity,
Yeah. So
so both of these you mentioned are triggered by activation of AHR, which is aryl hydrocarbon receptor.
yes.
They essentially bind to any aryl.
Any chemical.
I'm just wondering what would be the role of cadmium.
Okay, sorry, I forgot to mention one thing. Thanks for the question, actually. So here, I would like to point out one thing. So if you see in this pathway, we had identified the connectedness of AHR with cadmium based on our data integrative approach. But in the rest of the pathway, you see very less are connected until the end goal of our preeclampsia. So what we have further done is we have gone back to the literature and tried to link each and every key event in this process back to cadmium. So we were able to find experimental evidence from published literature to link that cadmium indeed causes each of these key events. So activation of AHR results in dimerization of AHR ART. Presence of cadmium can reduce the production of VEGF. We have found experimental evidence for that. Then going on, we have also found presence of cadmium causes abnormal placental labyrinth formation. And we also found cohort studies where cadmium was found people who are having increased exposure to cadmium also have increased incidence of preeclampsia from a cohort study in China.
what I was referring to is at the molecular level, how would cadmium, I mean, is it the presence of cadmium then leads to certain chemicals which then go on to activate these pathways? Because AHR, ARNT, for example, these are transcription factors which
Other pathways.
different things. Or is it cadmium itself which goes and does something? That's something which is not very clear. But yeah, maybe that's not required.
So AOPs are envisioned in a way such that if there is a single molecular trigger, we can track it until the end of the organism, until the organismal level. But maybe we can look deeper and see whether cadmium indeed induces each of these processes.
Okay. So final question from me. You don't have to answer it right now. We can take it over dinner. Couple of things. The first part where you had the literature survey and you did all that. Did you attempt trying LLM models to learn more beyond what you could figure out just by reading? That's one thing. You can actually do that. Secondly, the link database, right? You were using scaffold, but there was a paper that came out about
Links database.
okay, another like where you have scaffolds and you're learning from scaffolds and you're sort of clustering them, right? There was a paper that came out with where they use smiles as a linearized structure, and they learned a lot about. They developed a deep model which actually allows you to learn a lot more about how these would perform in different environments. So maybe that might be a easier way to see why for your remaining structures which you're not able to classify using links. The final question was in the like I don't want to use Lysol, but when you are associating vitiligo to a like a toxic chemical, have you seen any reports where people who are working in cleaning industry in US, I'm not talking about nobody studies, right?
Yes.
In US, where they were using these chemicals and there was a higher incidence of vitiligo in those individuals compared to normal population?
So let me answer the third question first. So indeed, we have come across various case study reports where medical practitioners and people who clean hospitals on a regular basis, they show increased incidence of this vitiligo on their arms and feet, which are more exposed to these chemicals. So indeed, we have found case study reports for that. Thank you very much.